So let's start with the vacuole now. Now the vacuole, they are non-cytoplasmic area present inside the cytoplasm and separated from it with the help of a specific membrane. So let's write, what are the vacuoles? They are non-cytoplasmic areas present in the cytoplasm and separated from the later with the help of specific membrane. अब ये जो वैक्यूल है, इनका ओरिजिन कहां से हुआ? The vacuoles, they are formed, they are originated from endoplasmic reticulum. अब ये जो vacuole है, on the basis of the contents and functions, they can be divided into the 4 categories, 4 types. अब ये जो 4 types का lysosome है, यहाँ पर हम discuss करेंगे. Now the 4 type of the vacuole which I will be discussing here, that is contractile vacuole, then sap vacuole, digestive vacuole and air vacuole, gas vacuole. Now here I will be dealing with the contractile vacuole. For the vacuole I am writing V here. Then sap vacuole. Digestive vacuole, then air vacuole और gas vacuole. तो ये 4 कैटेगरीज डिस्कस करेंगे. सबसे पहले स्टार्ट करते हैं कॉन्ट्रैक्टाइल वैक्यूल के अंदर. कॉन्ट्रैक्टाइल वैक्यूल जो है, इट इज प्रेजेंट इन द फ्रेश वाटर प्रोटोजोअन्स एंड एल्गी. तो पहले यहां पर हम डिस्कस करेंगे अकरेंस. Contractile vacuole, they are present in the freshwater protozoans and algae. So several examples about these organisms which contain the contractile vacuole that might be known to you. So in the protozoans, the contractile vacuole is present in amoeba, paramecium, and amongst the algae it is chlamydomonas. Now what is the function of the contractile vacuole? Contractile vacuole will maintain the osmolarity of the cell. Yes, it will maintain the osmolarity of the cell. So we can say one function which is associated with the contractile vacuole, that is osmoregulation. Osmoregulation के साथ साथ it is also involved in the excretion. Now coming to the sap vacuole, present in the plants as well as the animals. Plants के अंदर जो young stages है, जो young plant cell है, young plant cell के अंदर small sap vacuoles they are present. पर जो mature plant cell है, वहां पर ये vacuole fuse करेंगे and there is formation of large central vacuole which will cover 90% of the volume of cell. So you're getting this? So a plant cell के अंदर जब young stages है, young cell के अंदर present है small size vacuoles और mature plant cell के अंदर ये जो small size vacuole है, they will fuse, results in the formation of large central vacuole, and this large central vacuole, it will cover 90% of the volume of cell. अब ये जो sap vacuole है ना, इसके आसपास जो membrane present है, yes, the membrane which is present around the sap vacuole, this membrane, it is known as tonoplast. This is tonoplast. इस membrane के अंदर present है carriers, present है channels, and these channels, they are involved in the transport of ions and other material. Ions and other material, and this transport will occur against the concentration gradient. So you're getting this? जैसे ये sap vacuole है, ये sap vacuole की membrane है, that is tonoplast, right? तो tonoplast के अंदर present है कुछ carriers, कुछ channels which will allow the movement of the ions, which will allow the movement of the ions from the cytoplasm inside the vacuole. और ये जो movement होगी, that is against the concentration gradient. Against the concentration gradient, that is from low concentration to high concentration area, and for this movement there is requirement of energy, ATP is required. Now the sap vacuoles, they are involved in the storage. एक function क्या है, ये storage का काम करते हैं, like they are involved in the storage of amino acids. They possess sugars, even contain waste material, water soluble pigments, water soluble pigment like anthocyanin, which will give the color to the flowers and fruits, right? They will give the color to the flowers. Another, along with this, there is presence of esters. तो यहाँ पर जो sap vacuole है, उसका एक जो main function है, it is involved in the storage. Now coming to the digested vacuole. अभी मैंने यहाँ पर डिस्कस करा कि what is digestive vacuole? It is formed by the fusion of the primary lysosome with the phagosome. Yes. So this vacuole, it is formed by the fusion of the primary lysosome with phagosome. This digestive vacuole is present in protist, present in sponges, coelentrates, along with that higher animals के अंदर भी digestive vacuole present है. अब इस digestive vacuole के अंदर present है enzymes, and you very well know the function of the enzymes. They are involved in the digestion of food. They are involved in the digestion of food. That's why these digestive vacuoles, they are also known as food vacuole. तो इनके अंदर present है digestive enzymes. And these digestive enzymes, they are involved in the breakdown of food. Now coming to the gas vacuole, air vacuole. अब ये जो गैस वैक्यूल है, ये प्रेजेंट है प्रोकैरियोट्स के अंदर. इट इज प्रेजेंट इन दी बैक्टीरिया, एंड आई विल बी राइटिंग द एग्जांपल ऑफ वन बैक्टीरियल कैटेगरी वेयर दे आर प्रेजेंट, दैट इज साइनोबैक्टीरिया ब्लू ग्रीन alga. अब ये जो साइनोबैक्टीरिया है, इसके अंदर जो वैक्यूल है, इस वैक्यूल के अंदर प्रेजेंट है मेटाबॉलिक गैसेस, एंड दीज गैसेस दे विल रेगुलेट द buoyancy ऑफ द सेल. So you're getting this? They contain gases and this will regulate buoyancy of the cell. अब ये जो वैक्यूल है, इस वैक्यूल के आसपास प्रेजेंट है मेम्ब्रेन, एंड दैट मेम्ब्रेन is made up of protein. It is not a true membrane. So around the vacuole, the membrane which is present, that is proteinaceous. That is not made up of lipid bilayer. So it is not a true membrane. It is not a true membrane. That's why these gas vacuoles, they are also known as pseudo-vacuoles. Right? So this is all about the different components of the endomembrane system, where I had discussed endoplasmic reticulum, Golgi bodies, then lysosome and vacuole. So here I would like to introduce one term. That term is GERL, G-E-R-L. Yes, GERL, it is a complex. Yes, इस कॉम्प्लेक्स के अंदर क्या होगा, नाम से ही पता चल रहा है, it will contain endoplasmic reticulum, Golgi bodies and lysosome. So this is a complex which will be consisting of ER, Golgi bodies and lysosome. So this is all about the endomembrane system. Now I will be starting with the microbodies. Now let's start with the microbodies. Now microbodies, they are single membrane bound organelles. Single membrane hai, they have diameter ranging from 0.2 to 1.5 micrometer. This is the diameter of the microbodies. अब ये जो microbodies है, इनके अंदर होते हैं oxidation reactions. Yes, they are categorized for the oxidation reactions, but you please keep this in mind. यहाँ पर जो oxidation reaction होंगे, these reactions are the one which are not associated with the respiration. तो आपको मालूम है रेस्पिरेशन के अंदर भी ऑक्सीडेशन रिएक्शन होना है, बट हियर द ऑक्सीडेशन रिएक्शंस व्हिच विल अकर, दे आर द वन व्हिच आर नॉट एसोसिएटेड विद द रेस्पिरेशन. सो दीज ऑर्गेनेल्स, दे आर इन्वॉल्व्ड इन द ऑक्सीडेशन रिएक्शंस. अब माइक्रोबॉडीज के अंदर मैंने यहां पर 3 तरह की सेल ऑर्गेनेल्स पढ़ानी है. The 3 types of the microbodies I will be discussing here, that is peroxisomes, glyoxysomes and spherosome. So here I will be starting with these 3 cell organelles, and first of all I will be doing the comparative study between these 3 with respect to the some important features. So here I will be discussing peroxisomes, glyoxysomes, spherosomes. 3 microbodies. अब इसमें से सबसे पहले देखते हैं peroxisomes के बारे में. The peroxisomes, they were discovered by the scientist D. Dewey. Discovery is credited to D. Dewey. Glyoxysomes, they were discovered by the Breidenbach. Spherosome, the discovery is credited to the Perner. अब ये जो peroxisomes हैं, ये प्लांट सेल के अंदर भी प्रेजेंट होते हैं, एनिमल सेल के अंदर भी प्रेजेंट होते हैं. So the occurrence is in the animal as well as in the plant cell. earlier the view of the scientist was that कि ये जो peroxisome है ये सिर्फ animal cell के अंदर present है, but today we know they have the role to play both in the animal as well as plant cells. Glyoxysomes, they are present in plant cells. The spherosomes, they are present in plant cells. जो peroxisomes हैं, these are long-lived structures. These are long-lived cell organelles. But the glyoxysome, after performing their function, they will degenerate. After performing their functionality, after performing the function, they will degenerate. So you can say now they are short-lived or short lifespan. The same condition is there with respect to the spherosome. They are short-lived structures. After performing these functions, these structures, they are going to degenerate. So now here I will be discussing the important functions associated with these microbodies. तो सबसे पहले मैं peroxisomes के जो functions है यहाँ पर start करूंगी. So let's start with the functions of the peroxisomes. So we have to see here what functions they are performed by these organelles in the plant and animal cells. First start with the animal cell. In the animal cells, they are involved in the breakdown of amino acids, fatty acids, drugs, alcohol, toxins. So they are involved in the breakdown, in the metabolism of fatty acids, extra amino acids, alcohol, drugs, toxins, and the nitrogenous bases, that is purines. So you might be knowing the nitrogenous bases, they are of two types, that is the purines and the pyrimidines. The purines, they are double ring structure and pyrimidines, they are single ring structure. अब इस ब्रेकडाउन को परफॉर्म करने के लिए इन peroxisomes के अंदर प्रेजेंट है एंजाइम्स. So performing this breakdown, they have oxidase enzyme. तो कौन कौन से ऑक्सीडेज एंजाइम्स हैं जो इस सेल ऑर्गेनेली के साथ एसोसिएटेड है, वो यहां पर हम लिखेंगे. वन ऑक्सीडेज एंजाइम विच इज एसोसिएटेड विद दिस सेल ऑर्गेनेली, दैट इज अल्फा हाइड्रोक्सी एसिड ऑक्सीडेज. देन बीटा हाइड्रोक्सी एसिड ऑक्सीडेज. देन amino acid oxidase. Another one, that is urate oxidase. तो ये जो एंजाइम्स का मैंने यहां पर नाम लिखा है, these enzymes they are involved in the breakdown. When these enzymes they are involved in the breakdown, they release a byproduct. There is formation of a byproduct. The byproduct which is formed, that is H2O2, the formation of hydrogen peroxide. अब ये जो हाइड्रोजन पेरोक्साइड है, दिस केमिकल इज टॉक्सिक. ये एक टॉक्सिक केमिकल है. दिस केमिकल इट हैज द एबिलिटी टू कॉज द ब्रेकेज ऑफ द मेम्ब्रेन. इट कैन कॉज द ऑक्सीडेशन ऑफ द मेम्ब्रेन. तो ये जो टॉक्सिक केमिकल यहां पर फॉर्म हो रहा है, इस केमिकल का होगा ब्रेकडाउन. यस, ये जो H2O2 होगा इसका ब्रेकडाउन होगा विद द हेल्प ऑफ द एंजाइम विच इज अगेन एसोसिएटेड विद द peroxisome. द एंजाइम इज catalase enzyme involved in the breakdown of H2O2 और ब्रेक डाउन करने के बाद देयर विल बी फॉर्मेशन ऑफ द वाटर एंड ऑक्सीजन। तो अब ध्यान से देखिए यहां पर कि पेरोक्सिसोम्स के अंदर प्रेजेंट है ऑक्सीडेज एंजाइम। ये ऑक्सीडेज एंजाइम है, दे फॉर्म H2O2। पेरोक्सिसोम्स के अंदर प्रेजेंट है कैटालेज एंजाइम and the catalase enzyme is involved in the breakdown of H2O2। तो इसका मतलब पेरोक्सिसोम के अंदर there is formation of hydrogen peroxide as well as there is breakdown of hydrogen peroxide। It means this cell organelle, it is involved in the metabolism of peroxide। Yes, this cell organelle is involved in the metabolism of peroxide। That's why this cell organelle, what it is known as peroxisome। तो ये बात समझ आई, ब्रेकडाउन भी होगा, फॉर्मेशन भी होगा, मेटाबॉलिज्म हो रहा है पेरोक्सीसाइड का, पेरोक्साइड का। That's why these cell organelles, they are known as peroxisomes। Now here I would like to add one thing, that the oxidase is the smallest enzyme। Yes, this is smallest enzyme, and the catalase, this is largest enzyme। So we can say the smallest and the largest enzyme, they are associated with the peroxisomes, they are involved in the metabolism of H2O2। Let's see now what is the function of the peroxisome in the plant. In the plant, they are involved in a process that is photorespiration। Now what is respiration? It is known to all of you that respiration is taking in of oxygen and giving out of the carbon dioxide। तो यहां पर भी वही होगा, taking in of oxygen, giving out of the carbon dioxide, but now this respiration, it is dependent upon light। It is light dependent and only performed by the photosynthetic organs, green parts of the plant। So what is photorespiration? It is light dependent oxygen uptake and release of carbon dioxide by green parts of plant। अब इस फोटो रेस्पिरेशन को परफॉर्म करने के लिए देयर इज इन्वॉल्वमेंट ऑफ 3 सेल ऑर्गेनेलीज। फोटो रेस्पिरेशन को परफॉर्म करने के लिए 3 सेल ऑर्गेनेलीज की इन्वॉल्वमेंट है। पीसीएम, दैट इज पेरोक्सिसोम, क्लोरोप्लास्ट एंड माइटोकॉन्ड्रिया। Now you will learn more about the photorespiration when I will start with the chapter that is photosynthesis। So the functions of the peroxisomes in the plants and animals is very much clear to all of you। Now after this, now I will be dealing with the glyoxysomes। So let's start with the glyoxysomes। Now the glyoxysomes, they convert fats into carbohydrate। Glyoxysomes, they possess enzymes for the conversion of fat into sugars, fat into carbohydrate। तो देखिए ये फैट से शुगर्स बनाएंगे। तो अब देखिए ये जो शुगर की फॉर्मेशन हो रही है, अब ये फैट से हो रही है, किसी कार्बोहाइड्रेट सोर्स से नहीं हो रही है। तो जब शुगर की फॉर्मेशन किसी नॉन कार्बोहाइड्रेट सोर्स से होती है, जैसे फैट से यहां पर हो रही है, सो दिस प्रोसेस ऑफ द शुगर फॉर्मेशन इट इज नोन एज gluconeogenesis। If the formation of sugar it occurs from non-carbohydrate source, then it is known as gluconeogenesis। Gluconeogenesis को परफॉर्म करने के लिए जो पाथवे यहां पर इन्वॉल्व होगा, that is glyoxylate pathway. Glyoxylate cycle is involved. Now the conversion of the fat into the sugar, this is occurring in the fatty seeds like the seeds of castor and groundnut. कैस्टर और ग्राउंडनट के अंदर प्रेजेंट है फैट, और जब सीड की जर्मिनेशन होगी द फैट्स विल बी कन्वर्टेड इनटू द शुगर्स एंड दीज शुगर्स दे विल एक्ट लाइक द एनर्जी सोर्स फॉर द सीड जर्मिनेशन. नाउ द लास्ट वन, spherosomes. Now the spherosomes, they are involved in the storage and the synthesis of fats. Yes? So these microbodies, they are involved in synthesis and storage of fats. So it means these microbodies, they are also associated with the fatty seeds. Right? So this is all about the three types of the microbodies, that is peroxisome, glyoxysome and spherosome. Now I will be starting with the protein factories. And you know that. What are the protein factories? That is ribosomes.